Hello viewers, welcome once again to our ongoing series on genetics entitled Mendelism and Beyond. We have been taking some very in-depth studies on plant sex determination mechanisms, and today we wish to devote it to the third and the fourth part of the lecture series. As you know, learners, the plant sex determination can be environmental, and we have made a very detailed discussion. They could be chromosomal, which would now be subsequently discussed by us. And when we are talking about chromosomal systems, then we can have a whole range of combinations in terms of homomorphic chromosomes in the form of the heterogametic males, the XXXY mechanisms, the ZWZZ mechanisms, which is quite similar to the ones which we have already studied in the case of animal systems. In addition, we have the heteromorphic chromosomes, which would involve an active Y or the gene balance as we had in Drosophila, like the balance between the X chromosome and the autosomes. And then we have a vast array of different types of X chromosomes running the show of sex determination. Apart from it, the more important aspects in plant sex determination are the genic ones. They may involve the single locus as well as the multiple loci. If you remember, learners, the first part of the lecture, that is the first lecture in this series of sex determination in plants, was devoted to the environmental control of sex. So if I have just a little recap, we found that in the environmental sex determination, which was rightly abbreviated as ESD, the gender was not decided genetically. It was not fixed. It was actually decided after the conception, and it naturally depended on the environment. So that means most of the studies that we have done as far as the environmental control of sex determination is concerned has been restricted to the angiosperms, because they are naturally the dominant form of the ESD is in the form of the sexual lability or flexibility. So that means if there is a non-genetic cue, say for example like the availability of nutrients, or within a period of after fertilization there is a change in the photoperiod, there is a change in the nitrogen availability, naturally it may even lead to a change in the sexual behavior. Now this idea is in absolute contrast to the genotypic sex determination, because there, if you remember, the sex was established at fertilization, and it was the sex chromosomes which were responsible for determining the future course of the sex of an individual. The basic idea is that light intensity, photoperiod, temperature, as I said before, nutrient availability, and even the presence of certain hormones which are called as the attractants or the pheromones, which are emitted by the surrounding plants or animals, could also influence the sex of an individual. And mind you, this will happen during development or sexual maturation. If you remember, viewers, in our second part of the lecture on plant sex determination mechanisms, I had tried to make you familiar with some basic terminology that we were using while describing the nature of flowers in angiosperms. And if you remember, there were so many kinds of terms that we had enumerated. We had also stressed upon why the dioecious sexual systems in evolution are much more advantageous, because they are the ones which are responsible for the evolution of new species, and they would avoid inbreeding, and they would likewise avoid inbreeding depression. On your screens you can find the same picture which we had shown on the last turn, giving you different terms of hermaphrodites, monoeci, gynomonoeci, andromonoeci, and of course dioecious, gynandrodioecious, androdioecious, and of course I had asked you some questions on what a trioecious plant was. So the mastery of this terminology is a must to appreciate the patterns of evolution of sex among different types of organisms during the course of evolutionary history. Thereafter, we had begun with a discussion on the single gene sex determination, wherein we had learnt the details about the primitive sex determination in the unicellular alga Chlamydomonas, and I had told you that the sex in this case was determined by a single gene. This was quite common with Neurospora and yeast also. And viewers, there's absolutely no difference or little visible difference between the cells that reproduce asexually, say zoospores, or even the gametes in Chlamydomonas. So when they are not distinguishable from each other, and yet they represent different sexual forms. I would not say morphologically different sexes, they represent biochemical different forms. So they are rightly called as isogametes. On your screens you would find the same summary diagram with which we had made you familiar with how the isogametes of the two strains of Chlamydomonas, which are haploid, would fuse each other from their anterior ends, giving rise to a transitory quadriflagellate structure, and then finally giving rise to a zygote. The idea was that the empty plus or the empty gametes, howsoever identical they may appear morphologically, they are biochemically very different, because they would show clumping. MP plus would show clumping with empty minus, but empty plus and empty plus would never mate, and therefore they would not show clumping. The same was true with empty minus and empty minus coming together, showing no clumping, thereby proving that even though they were morphologically identical, biochemically they were different. And I had also reiterated some of the modern ideas that this particular locus has already been located on the chromosome number 6 in Chlamydomonas, and this is essential for the cell fusion in response to nitrogen depletion. So that means somewhere or the other the environmental sex determination control was also there. So to continue with the same discussion, let us see some of the very important examples of monogenic control of sex determination, and let us see how sex determination operates in asparagus. As you know, viewers, asparagus is a very important Ayurvedic herb. It belongs to the family Asparagaceae, and it is commonly called as shatavari or satamul. Now, this particular plant, asparagus, is a dioecious plant. Sometimes the female flowers may even show rudimentary anthers, and on the other hand, there are male flowers, they may show rudimentary pistils. So obviously, if the male flowers are going to show rudimentary pistils, they would never set seeds. Now, when such a plant is raised, we find that a male and a female plant, they are showing the segregation ratio of 3:1, which happens to be a typically Mendelian ratio. when these male plants were used to pollinate the female flowers on the female plants, again only 2/3 of them showed segregation, suggesting that sex in this case is controlled by a single gene. By the way, it also suggests that maleness happens to be dominant over femaleness. On your screens, you will see a chart where the real male plant, it is rare to have a capital P and small p, it is selfed. And after selfing, wherever we have the capital P, it is male. And therefore, the segregation ratio naturally comes to approximately 75% to 25% of males and females respectively. As you know, viewers, the genic sex determination is a little different from chromosomal sex determination, because the sex determination in major land plants, how did they evolve? So there must have been a major transition, as we know. So this evolution of sexual reproduction constituted a major transition in the evolution of life itself, because this transition must have occurred well before plants first invaded onto land. And this this event happened roughly 470 million years ago. And by that time, what were the ancestors of land plants showing? They had already differentiated male and female gametes. And that means spermatozoids were there, eggs were there, and different sex organs were also differentiated so as to produce them, that is the antheridia and the archegonia. So that means the first land plants, when they arrived, they had already evolved an alternation of haploid and diploid generations. That means the gametophytic and the sporophytic generations. So to say that in all the land plants, the differentiation of sex organs into eggs and sperms producing tissues, they would occur in the haploid or in the gametophyte phase. Now we can ask ourselves a very pertinent question. Which phase has the control over the decision? That means whether it is the gametophytic phase or the sporophytic one. And the second question, which is again very relevant, is that how the decision to determine sex is enacted. That means who is the controller and who is the transmitter to carry out this particular decision. Now, these two aspects would vary in different plant groups. As we know that the sex determining mechanisms are by themselves very diverse in non-vascular plants, that is in bryophytes, liverworts and mosses, and they would be slightly different in vascular non-seed producing plants like the lycophytes and monilophytes. And still other phase of departure would be in the clade of vascular seed bearing plants, which may include the extant gymnosperms and the flowering plants, of course, that is the evolution of angiosperms. Viewers, let us first begin with the sex determination in bryophytes, because they have recently some new information has been gathered, and it is believed that they show a UV system of sex determination, and they even have sex chromosomes. We know that in bryophytes, the gametophytes are either male or female, so we would like to call them as the dioecious species, because separate individuals are there and they are producing either antheridia or archegonia. So in bryophytic literature or in the literature of thallophytes, it is customary to call them as heterothalic. On the other hand, They could be hermaphroditic, which we would like to call them as monoecious species, where both Antheridia and Archegonia are present in the same individual. The biologists would like to call it as homothallic species. Now, here in this case, sex determination is a matter of differentiation of cells and tissues in different parts of the same individuals. And it is seen that in dioecious species, separate male and female gametophytes would germinate from spores of the same size. Remember the case of Chlamydomonas? Here the spores are of the same type. They are homosporous species, yet their sexual fates are different. So, sexual dimorphism nevertheless can sometimes be extreme also, because in some cases it is found that the male gametophyte is dwarf and it grows on larger female gametophytes. So, that means this type of variation may be there. But in many bryophytes, sex is determined in the gametophytes by the sex chromosomes, which we call as U and V. Now, these terms have been used by the bryophyte cytologists. If we look at an equivalence, so the U would be equivalent to what we think as the X chromosome, and V will be likened to the Y chromosome. So, this UV sex determination is prevalent in mosses, liverworts, hornworts like Anthoceros, Phaeoceros, Notothylus, Asperomyces, and this idea is also true for certain algae. So, then basically it is a haploid controlled systems, but then it is very interesting. Each of the individual, it is haploid, it has only one sex chromosome. Females are carrying only a single U and males are carrying a single V. So, it immediately gives us an idea of the X and Y at the back of our minds. But there is a big difference. Unlike the XY and the ZW systems, both the U and V chromosomes in this system are sex determining. In one case, it was only Y which was deciding. In this case, U also decides and so does the V. So, that means the haploid spores which are carrying U or a V will grow into the haploid gametophytes, but then their antheridiophores or the archegoniophores would depend on whether they have U or V, and union of eggs and sperm within these offspring will allow them to produce more spores. On your screens, viewers, you will find a summary of the UV system of this sex determination, which is very interesting, because a sporophyte undergoing meiosis would have two types of spores, one carrying V and the other U, and their gametophytes would be different, and then U would be giving rise to an egg, V would be giving rise to a sperm, and fertilization would result in again the formation of a zygote. Chromosome mechanisms in Markensia polymorpha, which is a liverwort, have been carried out by using modern techniques, and you know that Markensia has a very small genome, say about 280 MB. Whereas you remember I told you that this MB means a megabase, which is a length for the DNA fragments, and it is considered to be equivalent to 1 billion nucleotides. And transforming it into cytogenetical language, this would be equivalent to 1 centimorgan. So, Markensia has 8 autosomes and 1 sex chromosome. So, that means total N is equal to 9. So males would have, as you can see on your screens, males would have 8A and one one Y, which means no X. Y is smaller in size than the X chromosome. So far so good as far as the human genetics is concerned, because here also the Y is the smaller one. Females again would have 8 autosomes and one X, a large one, but then it would have no Y. And the diploid sporophyte would have X and Y. So it's very interesting to know that there is no recombination between the X-linked genes, because there are no homologs. On your screens you would find a pictorial depiction of the entheridiophores and the archegoniophores of Markensia polymorpha. We take the case of another liverwort, which is Spherocarpus. Learners, you must remember that the sex chromosomes was seen for the first time in Spherocarpus by Ellen in as early as in 1917. Now, this also shows somewhat XY type of mechanism. The haploid number is 8, which means 7 autosomes plus an X or Y. When it is heterogametic, that means X is larger and Y is even smaller than the autosomes. On your screens you could find a pictorial depiction of the life cycle, where the haploid spores are fusing with each other, giving rise to two different thalli, the male and the female one, which are having the entheridiophores and the archegoniophores respectively. And upon the fusion of sperm and the egg, a sporophyte is formed, giving rise to a diploid individual. So that means this type of mechanisms have also been now translated into modern studies in flowering plants. And as you know, the model system of Arabidopsis thaliana has given us lots and lots of information, where the specification of male and the female sporophytic floral organs, that is the stamens and pistils, by the ABC genes has been elucidated. Viewers, we will have an opportunity to talk about the ABC model of floral development and sexual control in one of our subsequent lectures. So one plausible hypothesis that we can carry is that the sexual differentiation of the sporophyte is imposed upon the retained gametophytes which are developing within the sporophytic tissues, and it will go ahead. So therefore, during land plant evolution, what would have happened would be, or would have been, that sexual differentiation has shifted from a purely gametophytic program, because all of them were showing a haploid cycle, to a situation where the sporophyte now controls the sexual differentiation of the gametophyte. The question now arises, are there regulators of gametophytic sexual differentiation that are conserved even across the land plants? There is a gene, an autosomal gene, which has now been recognized with the help of modern genetical techniques and molecular markers, and this is called as MPFGMYB gene. Now this gene is known to promote female sexual differentiation in the liverwort Markensia polymorpha. It is very interesting to know, viewers, that this particular gene belongs to the MYB subfamily, whose members are known to regulate the female gametophyte development in the angiosperm, Arabidopsis thaliana. So now it's a matter of real excitement that the same gene which we find, or at least the same family of gene which we find in a primitive plant like Markentia, is also occurring in Arabidopsis. On your screens you can find a very interesting picture which summarizes the origin of maleness and the femaleness. As you can see, the FGMYB gene is being controlled by an antisense gene which is SUF, and when SUF is present, then this gene is not working, and this is a default, and as a result you will find the male being produced. The X chromosome feminizing will take place only when this particular SUF or the antisense is not present, and then only the X chromosomal feminizing will take place. That is, this gene will be active, the FGMYB gene will be active, and then it would mean a production of X. So that means this particular MY type of gene is conserved, and since it is present in such diverse groups, we would conclude that it has a shared origin of gamete formation in plants. So that means this whole idea tells us that there is a great diversity, yet these particular genes are conserved. Of course, the situations are different in angiosperms, because now the female and the male gametes have been highly reduced to a 7-celled embryo sac and a 3-celled pollen grain respectively. But then the sex specific differentiation is dependent upon the sporophytic generation and not on the gametophytic generation as it was in its predecessors. With this little idea, we would take up the discussion further in our next lecture as to how the different type of plants, again, they are responsible for controlling the sex determination or the sex in plants. Thank you.